All a microscope is a really powerful magnifying glass.
The great advantage of the light microscope over the electron microscope is the fact that you can zoom into the cell, you can focus into the cell without harming it.
So because of that, you could look at the natural function of the cell in real time.
So what was the problem? The problem is if you focus down the light to a point, you will have a diffraction blur.
The lens diffracts the light, and as a result, you can't focus the beam to anything smaller than the wavelength, approximate wavelength of the light itself. So that means that if you are hoping to look at an object that's less than the wavelength of light, you do not have a resolution that is smaller than about 200 to 400 nanometers. Now, it turns out that molecules are about 3 nanometers in size. So the problem is, if you want to look at individual molecules, all you will see with diffraction-limited imaging is big blobs of about 200 nanometer, blurry spots.
Of course, I knew that it was not possible to focus the light more sharply than about half the wavelengths of light, but it doesn't mean that it's not possible to generate, say, spots of excited molecules that are much sharper than that. And one way of doing it is to accept that large area of excitation, but then use another beam of light that switches off the molecules at the outer part of that area of excitation. And if you do that, you end up with a region that is much smaller than the initial region, and therefore you have a much smaller spot of fluorescence. And if you have a much smaller spot of fluorescence, you can use that to take sharper images. You just scan that spot across the specimen, and in the end you will see more details, because the sharper the spot, the more details you get.
The fundamental principles of these two imaging methods are similar, or I would even say the same, because they are both taking advantage of the fact that the molecules are switchable.
You could take the blurry spot given off by an individual molecule, and if you could switch off that spot, you could then fit the spot using mathematical equations such that you could determine the center of that spot. And that center is where the molecule is.
So our microscope uses laser light for illumination, and we actually have several different colors from violet all the way to red for illuminating the sample. We have a fast shutter system that is able to switch the laser light on and off very quickly in order to activate and deactivate the switchable fluorophores.
Of course, in many biological samples, molecules are so densely packed so their images overlap, and then you lost the ability to fit this single molecule image to find the center. So then we thought hard about the problem, then we came up with this trick.
The trick is you switch on these molecules separately in small groups, where each of the molecules that have been switched on are far enough apart so that their spots don't overlap.
Eventually, I get a sufficient number of molecular positions, and then I stack them together to form an image.
So this is the storm image that we generated from all of our individual localizations. And in this sample, you're looking at microtubules in a cell. If you zoom in really closely, you can see that, at its most basic level, the image is just made up of a bunch of locations. And then, when you zoom out on all of these crosses, there's millions of crosses. I can compare this image to the conventional resolution image. And you can see that in the conventional resolution, you can't make out the individual microtubules, but in the storm image, they're easy to resolve.
For the first time, you just saw this absolutely spectacular distribution of tens of thousands of molecules inside the mitochondria. And when I saw that, I was ready to sort of fall on the floor.
You can see now details, for example, structures in the mitochondrion, or structure in the nucleus, or say, even, for example, a neuron to see what it's doing when it's learning. I think there's a whole world down there to be discovered with focused visible light.
This is exactly why I loved microscopy, because people say that seeing is believing, right? I saw it, I said, this thing, has a future.